Before we take a bite, before we feel hunger, and long before we consciously decide what to eat, our gut has already begun steering the outcome. This is the domain of neuropods, specialized sensory cells in the intestinal lining that act as a kind of internal surveillance system, constantly reading microbial cues and nutrient content, and relaying that information to the brain. Neuropods don't wait for hormonal signals to slowly circulate. They use rapid, synapse-like communication via the vagus nerve, reaching the brain in milliseconds. And the messages they carry have startling implications for eating behavior, particularly our preferences, cravings, and feelings of satisfaction. The emerging science around neuropods paints a picture of the gut not as a passive processor of food, but as an active sensory organ, one that evaluates every meal in real time and shapes behavior accordingly. These cells resemble taste buds, but are embedded deep in the lining of the small intestine. Unlike taste buds, which detect external stimuli on the tongue, neuropods sense internal content, sugars, amino acids, fats, and even microbial metabolites produced during digestion. What makes them revolutionary is their ability to form direct connections to neurons, especially afferent fibers of the vagus nerve, allowing instant feedback from the gut to the brainstem and eventually to cortical areas involved in decision-making. This means that what we think of as food preferences or cravings are often preloaded responses, shaped not by conscious thought, but by what our gut microbiota are producing, consuming, or requesting. When neuropods detect specific bacterial metabolites, like short-chain fatty acids, SCFAs, from fiber fermentation or indole derivatives from tryptophan metabolism, they adjust their signaling accordingly. Some signals encourage satiety and reduced intake, others may increase cravings for high-energy foods. This microbial whispering into our gut's neural circuits can feel like an urge or impulse, but it's really a highly sophisticated feedback loop. Consider the example of sugar. Studies have shown that neuropods can distinguish caloric sugars, like glucose, from non-caloric sweeteners, like sucralose, not by taste, but by post-ingestive value. Only real sugars activate neuropod pathways that signal reward and satisfaction to the brain. This explains why diet sodas may taste sweet but fail to reduce cravings. They don't activate the gut-brain reinforcement loop. Similarly, fat detection in the intestine, particularly long-chain fatty acids, prompts neuropods to release neuroactive substances like CCK, cholecystokinin, which slows gastric emptying and promotes fullness. But the efficiency of this signal depends on the gut's microbial composition and the health of the epithelial neural interface. The role of microbes here is crucial, not just as participants in digestion, but as informational agents. Some bacteria can amplify satiety signals by producing SCFAs that encourage the release of GLP-1 and PYY, hormones that make us feel full. Others, through lipopolysaccharide LPS, or excessive flagellin expression, can trigger low-grade inflammation, disrupting neuropod function and leading to impaired satiety perception, a phenomenon seen in obesity and metabolic syndrome. The gut becomes deaf to its own satiety cues, leading to prolonged eating and poor feedback control. In animal models, when neuropod vagus circuits are artificially disrupted, feeding behavior becomes erratic. Mice fail to regulate intake based on nutrient quality, overeating or undereating indiscriminately. Conversely, stimulating these circuits can reduce compulsive eating, even in high-fat, high-sugar environments. These findings strongly suggest that neuropods are not just sensors, but gatekeepers of food motivation, able to tilt decisions toward or away from certain foods based on a mix of microbial, nutritional, and emotional context. What's especially compelling is that all this happens before conscious awareness. By the time we think we're choosing a salad over fries, the gut has already weighed in, modulating dopamine release, influencing hypothalamic signaling, and adjusting our perceived level of need or reward. In this sense, our brain doesn't make food decisions in isolation. It acts on data already curated by the gut's sensory infrastructure. From a therapeutic standpoint, this offers enormous potential. Rather than relying solely on willpower to change eating habits, we might reprogram gut-brain circuits by enhancing neuropod function and optimizing microbial inputs. Diets rich in fermentable fiber, polyphenol, and fermented foods can boost beneficial microbial activity, leading to better satiety signaling and healthier decision-making. Similarly, targeting neuropod sensitivity through next-generation smart probiotics or neuromodulators may offer a new frontier in treating obesity, binge eating, and even mood disorders linked to food. In conclusion, Neuropods are the gut's hidden sensors, an interface where microbes, nutrients, and neural circuits converge to guide behavior in ways we're only beginning to understand. They remind us that eating is not just about choice or culture, it's a deeply biological act, constantly shaped by organisms and cells operating beneath consciousness. Far from being mindless impulses, our food decisions may be among the most intricate and pre-scripted behaviors we perform, choreographed by a team of cells and microbes that have been learning what we need since the day we were born.